Hello, everyone. Hello, everyone. My name is Menachem Moshelion. I'm from the Hebrew University of Jerusalem. I'm a plant physiologist. And I would like to talk to you today about hydraulic conductance. Let's give you some introduction. I prepared a long one. So this is the highways of the leaf, right? This is our Rhodopsis leaf. It's a plant, promising. And you can see that in 1 centimeter, you have approximately 20 centimeters of veins. That means that the leaves actually need to move all this water through the plant to the cells, to the mesophiles in particular. But the funny thing is that when the plant is under stress, we have reduction in transpiration, of course, right? This is a Rhodopsis drought. So you see after 4 days, you have about 50% stomatal closure. But when you look on the hydraulic conductance, you see that after 2 days, you have 50% of hydraulic conductance. So hydraulic conductance is much faster than stomatal conductance, and you can also see it in one of Lauren's papers. You can see this is the same leaf. When you measure stomatal conductance, it's much slower than hydraulic conductance of the same leaf. So the liquids stop moving in the leaf much faster before the stomatal closing, and this is not embolism because it's recovered immediately. So what is it that actually controls the water flux in the leaf? We think it's the bundle sheet cells. These parenchymatic cells that actually surrounding every xylem you can check. We think that the biological control, mainly a component of these cells, actually controlling the fluxes out of the leaf. You can see these cells always. This is a very tight, nice bundle sheet. You can see it in any tissue. Look, this is from our Rhodopsis. You can see how nice over the xylem, this tomato. We always see these very, very tight living cells around the dead xylem. We all know the xylem is dead. But I suggest these living cells are actually the membranes of the xylem. Now we try to convince. So what is K-leaf? I'm going to use it a lot. This is the hydraulic conduction. Basically, it's how much water, not gas, liquid water, passing the tissues. So basically what you need to measure is transpiration, the water potential of the leaf, and K-leaf is actually water per water potential, how much water can go via or through a megapascal. Okay? So, and of course, you need to separate it from the gas exchange. Okay. So what do we know about the selectivity of this bundle sheet? So actually, during the last 2 decades, we have many papers that suggesting that, for example, if you feed the leaf, this is the work from Amnon Schwartz lab, with potassium chloride, the potassium will go out from the isotop, but if you feed the same leaf with potassium, the potassium will be 99% absorbed. So similar salt, one of them totally dripped out of the leaf, and one of them totally absorbed. This is not passive, this has to be active. Moreover, in my lab, we show that there are aquaporins on this tissue that actually control the water fluxes. And when you have ABA, actually these aquaporins are blocked, so we can see very nice shift in the bundle sheet water regulation on stress level. So when you have ABA, the amount of aquaporin reduction, and you have much less flux out of the xylem. All of this published, so I'm just running through. So it's going to be a long input. So what we suggested that when you have ABA going in the xylem, you know, many times under stress, ABA goes in the xylem, the ABA actually reduces the osmotic water permeability of the bundle sheet. So you have less water flux out. We actually prove it. We can extract the protoplast and measure it. And then you have low water potential and relative water content because it takes time until the stomata close, right? So it's a balance. So in the end, the stomata will close and respond to these changes in the intermediate parts between the veins and the gouges. How did we prove it? We could label the bundle sheet using a GFP. We can extract the protoplast and we can measure the osmotic water permeability, but we could also extract protein and genes out of it. And we did, when we did, Noah Vigorda was a former PhD student in my lab. When she did it, we actually find the whole blue light signal transduction pathway expressed in the bundle sheet. So this is a very important signal transduction pathway. You know, there are blue light sensors, spots, that sense the blue light, actually signal the AHA pump to push pump outside of the membrane, and also to depolarize the membrane, which actually activate channels and insert water. We actually have the same exact mechanism in bundle sheet. And the question, what is this mechanism doing in the bundle sheet? So we know that under stress, the pH become more acid in the xylem. So one of the questions we asked, can it be that under stress, the pump, the H pump, actually controlling the acidity of the xylem? And Yael Grunwald, my former PhD, this is exactly what she did. So she could actually use dyes to dye the xylem pH and measure it. And she actually proved using mutant and pharmacology that the pumps, the H pump, actually regulating the flux of protons into the xylem, which makes it about 5.7 under well-irrigated condition. But under stress, when you have ABA, this pump is actually blocked and you have alkalinization. So again, you have a membrane that controls the fluxes of the xylem. The xylem is not really there. Now, we also know that under blue light, there are many papers, I just brought one, blue light dramatically shift very fast the hydraulic conductance of plants, right? So we thought, hey, maybe this is the same signal conduction pathway starting from the hot ones. And Sanbon, Gorsa, and Yael actually together work on this question. We find all the genes they're expressing. And when we knock out the hot ones, using, again, I'm not going through the details, but when we knock out the hot ones and we use blockers and use red light and blue light, we could prove that actually the blue light activating the hot one in the bundle sheets, which actually activate the AHA2, and this changed the polarization of the membrane. The pH in the xylem actually activating the aquaporins, and this increased the water flux through the membrane, and thus increased the water permeability. This is what under well irrigation condition. Under stress, by the way, this was published. If you want more data, this is why I'm running so fast. So under stress, the question, how come ABA block this mechanism? And the question is, we know the ABA sensors are there, they are expressed there. So we asked ourselves, what is the exact mechanism that block the aquaporin? Because we know that when you put ABA, aquaporin are blocked almost like in 2 to 3 minutes. Fast reduction of water permeability in the protoplast level. So it's very, very fast. So Tanmay Thorn, a former PhD, together with Professor Nava Moran, we went to ask this question. Actually, we said, okay, we have all the genes, so maybe it's actually similar mechanism working in the bundle sheet. Till now, it was the same. Everything we checked was very similar. Actually, it was not identical, like FOT1 is in the bundle sheet and FOT2 is in the guard cells. But these are basically the changes, not dramatic one. So the idea was that maybe IBA starts signal transduction pathway increasing calcium, which in blocking the AHA, changing the pH in the cytoplasm and outside the cell, and also doing the depolarization changes. This is exactly what we did. Again, we work with protoplast only of the bundle sheet. So this is a big advantage. You can actually extract cells from the leaf, but you know you work only on the bundle sheet. We use a microscopic system that you can either put blue or red or just red, so we can really shift the light very fast and very easily. We work with guys that can measure either the membrane potential, because you know when you block a pump, you do two things. You change the polarity by the fact that no proton going out, also you have change in polarity, but you also change the pH. So we had the ANEPS, which actually measure the membrane potential. We had a pH measuring the pH in the xylem, so it's the apoplastic pH. We use a patch clamp technique to measure currents of ions. And we use blockers. So, for example, we use the BAPTA that actually block calcium. We had BAPTA that work either in the cytoplasmatic. So when you put the BAPTA AM, that means you don't have calcium in this cytoplasm. Or we use a BAPTA which is external. So we can either block the calcium outside or inside cell. And we could inhibit the AHA proton pump, calcium channels, and also the proton pump of the vacuum. Okay? These are the materials and methods. And again, it was not fully published, but part of it is already in bioarchive if you want. So what we found is that ABA actually activates calcium channels that increase the calcium in the cytoplasm. This calcium level actually activating a vacuole pump. This pump actually controlling the cytoplasmatic pH to stay 7. It was very surprisingly because we thought that the cytoplasmatic is going to buffer, but it was not due to the pump. And we are not sure, but it might be both the pH and the calcium actually are the one that block the AHA. And of course, when the AHA is blocking, immediately you have a change in pH, but also, and then this actually blocks the aquaporins and reduces the PF. So this is nice, but also you have a depolarization. And using a patch clamp, we found that this depolarization actually activating a potassium channel, a depolarization potassium channel, that enable a potassium flux, again, just like you have in the guard cells. This was the big question. Why would a bundle sheet need to release potassium, probably either to the xylem or to the phloem? We don't know. But there is a clearly release. We could prove this release by feeding leaves done by Veronica, master student in my lab. So she feed the leaf with or without ABA, and she took the the extract, and clearly there is more potassium when you feed leaf with ABA. So there is a release of potassium. So why would a plant respond to higher potassium? I need to run, so we took a potassium mutant plant, and we saw that the potassium mutant plants actually under stress. This is a score. This channel is external channel. So this plant does not release potassium to the xylem flux, and it's only also maintain higher yield. Very significant. So the potassium mutant that cannot release potassium to the xylem maintain higher hydraulic conductance under stress. Funny. We decided to measure the whole plant. I'm a big fan of whole plant transpiration. To me, this is like the most important trait when you need to measure the plant response. So for me, the transpiration is like the last resort. You can actually measure absolute data. So we use the plant array system. So the idea that you can put as many plants as you want simultaneously, and you can measure simultaneously the soil, the plants, and the atmosphere. So all the plants are exposed to the same structure. You can do a truly randomized, and each plant getting its own irrigation by a feedback system. And all these measurements are absolute. So it's a real measurement of how much water the plant really loses. Okay? This was published, so I'm not going to get into details, but all of this measurement of the whole plant transpiration, water use efficiency, stomatal conductance, we measure stomatal conductance to the whole canopy. So we control the drought and we can measure it. And of course, we put the mutants versus the wild types. We expose them to drought. One of the nice things with this system, you can do standardized drought, which means all the plants exposed to the exact same drought, regardless of the amount of transpiration they do. A very critical thing, because if you just close your tab when you do drought, each plant going to drought differently. And then you cannot really compare them. It's different factors, right? So the system enables you using these valves it has to control each plant separately. So you have a very similar drought. And here what you see, that the potassium mutant really keep more transpiration under the same drought. So really this mutation going to make them to survive probably less, right? Because they don't release potassium to the xylem, and as a result, some signal transduction that we're not sure of what, keep transpiration higher. You can check it also in the momentarily minute, so you can take the daily transpiration, this is by the minute. Clear, when the plant is under drought, this is the control, no difference. And under drought, a very clear difference, which plants are behind the wilting point, but still they open stomata higher. So something with the potassium release to the xylem actually we think is a very important regulator to control the K-leaf, but also the whole plant transpiration. So my take-home message to you, xylem might be dead, but they are covered. Remember the bundle sheet, they are living cells, and I think they have a tremendous impact on the plant's stress sensitivity.
One question? Ian.
Thank you very much for the nice presentation. You looked into rewatering the plants, the potassium deficiencies.
They actually recover the same. I think we didn't do enough drought. I mean, you saw they are totally wilted, but we see it all the time. Actually, we see a fully wilting. We do recovery. The recovery is very nice because you can do the recovery instantaneously to all of them, and we measure the slope of the recovery. So I guess we didn't wait enough because all of them recovered and we didn't see anything significant. Usually when you see problem with drought response, you see a very nice difference in the recovery. Here we didn't. So I'm still not sure even if this potassium goes to the xylem or to the phloem. So I know it's going out, but I'm not sure where.
Hello. I have 12 minutes, so I decided that I should do something playful. I don't have much time. I'm going to talk about somatal optimization under uncertain climates, and I will start, obviously, with Super Mario Kart. For you who were born in the wrong generation, Mario is one of the most famous video games ever. And why do I want to talk? Well, let's talk a little bit. I promise it will connect to climate eventually. So Mario Kart, Mario drives in a track, and many players compete to get first. So here we see Mario driving, and there's the coin. Of course, Mario wants the coins, and we see there's a banana peel. This is bad. There's a turtle shell. This is bad. So here we see, oh, let me close this. I don't like this. Yes. Second. Okay. And so here we see, where's the laser here? Right? Yes. So those two top things are good things that Mario needs to get, and those two bottom things are bad things. Okay. There's not only Mario, you can choose many players. So here we have Donkey Kong, and each one has different traits, different acceleration, top speeds, weights, etc. They behave really differently. If you play the game, you know it. And finally, they compete to know who will get there first. So the criterion is a minimal time. And whoever gets first gets the trophy. Okay, so let's translate that to another language here. So I have Mario, or you, the player. You are an intelligent agent. You or Mario. Let's talk about Mario. Mario perceives its environment. He takes actions autonomously in order to achieve goals. And Mario may improve its performance with learning, or he may use knowledge. So I just translated the video game into a different language. And this language, okay, and then let's translate that to a picture. So I have an agent, and the agent perceives its environment. And based on that, the player can take actions, real life actions, to accomplish some goal. Here, the goal is minimal time. Unfortunately, at a certain point, I need to introduce plants into the mix. So now I want to claim that my plants are my agents. And the agents also have an environment that they sense. There might be good things like solar radiation and water, or bad things like high temperatures or dry soil. And they can even perceive, so they perceive this environment, and they can even perceive their internal state. They might be thirsty, and they take actions in real life. So what are the actions? Here we see the stomata, the pores in the leaves. And by opening and shutting down those stomata, they can achieve a goal. Here, it's an assumption that what the plant wants to do is to maximize its carbon assimilation, photosynthesis. The same as before. Now we have the plant instead of Mario. The plant is an intelligent agent, perceives the environment, etc. All this language is the language natural to the following fields: artificial intelligence, machine learning, reinforcement learning, optimal control theory. And those are the lenses through which I look at the plant. Now, what do I care about? I care about plant traits, such as water use efficiency, resilience to drought, and the plant's risk policy. Okay, so we have a few strategies that the plant, now whenever I say, it's hard to talk about the plants and show just a picture of a plant driving a cart, so we'll have Mario standing for the plant. the plant can drive at full throttle, so maximum speed. There's only here and now. Tomorrow, who cares? Drive as fast as you can. And so here we have a carbon simulation in red and water transpiration in blue, and we have soil moisture. And then finally, I can translate that into something that the plant wants to optimize. It wants to optimize carbon simulation while minimizing water losses. And so whenever I optimize this H thing here, then the plant will be doing its thing. We have a lambda, which is the relative price of water in carbon units. So that's the water use efficiency. And the control is the stomatal opening is stomatal conductance is the control. So what actually the plant does is changes that. Okay, so let me bring in Menachem's talk from before. I don't care if it's stomatal conductance or any other conductance, a conductance. So here I will say just stomatal conductance. The plant changes that to such that this quantity is maximum. And there's no tomorrow. So it instantaneously optimizes every second, every millisecond. The plant wants to just maximize this function. Thanks to Menachem, he showed his setup from before. We got data from him, and we have experiments with many tomato lines, different variants of tomato, going through a 12-day dry down. And here are some of the results for one of them. So here you see data in gray, and we have two different variants of the model that I didn't show many details, but it's not too hard to make the model fit the data, kinda, right? So in the first days so here we have soil moisture, it's kind of high at the beginning and then goes down, and you have those things going up. It's not we don't cut water. Menachem doesn't cut water instantaneously. It gives a little bit, otherwise the plant would die in 3 days. So we can see that water content goes down, and we can see how transpiration varies throughout, and we can simulate what is the stomatal control and the water use efficiency. On a daily scale, the model, which is minimal, I didn't show it, but it is the very simplest model we could think of. It does a kind of okay job in simulating that, and from that we convinced ourselves the following: that tomatoes, the assumption that tomatoes instantaneously optimize their carbon gains is consistent with what we measured. We think that in general, crop plants, they have this driving at full throttle. They are taken care of, usually with irrigation. So the plants can do their maximum and just drive as fast as they can. One of the parameters of the model is the vulnerability to dry soil, and we have different tomato lines, and they have different vulnerabilities to dry soil. And we hope to get a really clean message. Okay, this line is more resistant to drought, but we did not, sadly. Why? Because we have an obvious surprise. They are extensive and intensive quantities. What are extensive quantities? It thinks that the absolute value is important. The size of the pot or the total leaf area is so much more important than other parameters of the model. We have a model we did not create it. We adapted from other people, and it has many parameters that talk about the photosynthetic chemistry, photosynthesis. Those parameters are not really important compared to the pot size and the leaf area, and that drowns the signal. So unless we do a very, very controlled, and Menachem's experiment is awesome. I think it's the best data I've seen. And still, if we don't control really some such things such as pot size and leaf area, the signal is drowned. Okay, so I showed you the first strategy. Let me show you a second strategy that we can think of, which is not just here and now. So that's the first strategy, instantaneous maximization of carbon simulation, but it depletes soil moisture very fast because the plant, there's no tomorrow. I don't care. I'll do whatever it takes. What else can we do? We can assume that instead of instantaneously optimizing carbon assimilation, we can take some interval over time. So the plant thinks about the future, and over a certain period, say a 12-day dry down, the plant will say, okay, I'm not going to use all the water today. I'll keep some for tomorrow because I don't want to die too soon. So that's the second approach. And this approach is been around for 50 years, widely adopted. So let's see, Mario, for the plant. Mario needs to, the plant needs to do its thing. So under the first strategy, when we have instantaneous maximization, all that the plant cares is every instant in time to get the maximum gain, but it might lead the plant to death. This is in the video game, this is death. All right. The other strategy is that I think about the future, and then I can trace a better path that hopefully doesn't take me to death. So that's the second strategy. And there is a recent paper that shows that this strategy is better, on average, is better than the first, the instantaneous optimization. But there is environment, and the environment can be random, right? I don't know when there's going to be a heat wave or, I don't know, a rain. There's uncertainty regarding the future. So what do we do with that? With uncertainty, what happens here is that Mario thinks about the whole future as Mario knew the future. Or the plant. But the plant doesn't know the future. The plant assumes things about the future and has to do its best. So the best under uncertain conditions, thank you, is to think that the present moment is very important. I'll take that into consideration. As time goes by, the future is less and less important. My actions now should not depend on what will be next month. If I can't predict what will be the weather in 4 weeks from now. So how do you do that? So this future is uncertain. So the present is more important than the future. And that what do? We introduce something called a discount factor. It says that, well, the present is more important. We do that all the time. Not only I'm assuming plants do that, but everyone does that. And that comes from economics, but everywhere we see that. So what is this discount? Is akin to a time horizon. How far can the plant see? So we have such a function, and if we have a discount rate rho, it goes to infinity, then we see that. So the time horizon is super, it shrinks and shrinks and shrinks, and we get to the first approach. There's only here and now. That's the first strategy. And when the discount rate goes to 0, then it gets flatter and flatter until it becomes 1. So when this is 0, then the whole exponential is 1. So that says that the future is as important as the present. There's no difference between them. So we are using this approach. And so when we have a short time horizon, we are talking about plants that take a lot of risks, and I associate that with anisohydric behavior. Anisohydric is when leaves don't close their stomata readily, and so the water potential inside the leaves can fluctuate a lot. When we have a long time horizon, so the present the future is as important as the present. So this is the plant being more risk averse, and that's associated with isohydric behavior. And that's not a binary choice. It's a spectrum. I can go from, usually I have in my manual, if I want to model plants like in this framework, I have to choose either that or the other. I'm putting everyone in the same spectrum with this discount rate, and using the same thinking of machine learning, I can. There are other axes. One of them is exploration, exploitation. How risky I want to be. It's different from discount rate. So how much I want to probe my environment and take risks. And I want to introduce that also to describing plant policies. And again, policy is a big word there. So take home message. I'm the guy who chose to talk about Mario today. In 6 months time, 6 months, you'll remember that hopefully. And with some luck, you will also remember that all that the language that I've used today, it comes from reinforcement learning. And this, I'm claiming this is the natural language to make sense of optimal somatic control, because we've been doing that all along for 50 years, but I'm framing that in a larger context. Thank you.
One question, Mario?
So you adopt ideas from economics and policy, which are based on intelligence planning, which
Yes.
plans don't have.
Right.
So the only strategy that can work is the only solution is that if one of these strategies leads to seed and propagation and survival. Otherwise, I don't know how the plan can.
Okay, let me go deep into philosophy. I don't believe in free will, so we're all machines. But using the language of machine learning, where is intelligent agent? Somewhere in the path here. This intelligent agent and everything. Intelligent, we should not be fooled by this word. A thermostat addresses all of those. We've built machines, and a thermostat that we have in the air conditioning or whatever is all of those things. So I don't have assumed intelligence in our sense, in our animal sense. Anything goes. We build machines that are already intelligent in this sense of the word.
If I may, I just want to continue what Dan said. Evolution. So you're assuming the plant goal is to maximize photosynthesis, but for this to be main plant, you need to show that photosynthesis correlates with fruits and with reproduction.
Yes.
Because plants want to reproduce; they don't want to produce photosynthesis.
Right. Yeah. I don't care about. Actually, I agree with you. I care about fitness. Let's talk about fitness.
Fitness.
Yes, I agree. And I think this is the better language. So using this framework, I want to play. So I have a goal function, right? Something that I want to maximize. I show there. There's an assumption that the plant wants to maximize carbon. I don't know that to be true, and easily I can change that rule for any other rule and see what works best. For 50 years, we've been assuming that plants wants to maximize carbon assimilation while minimizing water losses.
What about annual and perennial plants? You're on annual and perennial plants.
I'll be around today and tomorrow. Okay. I don't know. You're the
You must.
I want to talk about plants policies. Those are because I'm reading a lot about isohydric and isohydric. Is varying also between the species, within the plants. So there are plants forced to have one strategy, other, or they can change during the cycle.
Oh, is that a question? I have no idea.
That's a question.
Yes, I don't know. I'm not a plant guy.
Good.
Yes, I am hopefully trying to bridge between two different. I know neither, and I want to bridge between them. Yes. Okay.
Okay, thanks everybody for staying. And I hope I won't keep you from your lunch too much. And I wonder, is Yair still here? Great, thanks. I want to add to the discussion of strategies, what I learned in ecology when I was a student of R and K strategies and competition. And I recall a lecture of Missy Holbrook, introductory lecture. She said the reason that wood came into being and all the hydraulics that we're talking about is for competition, because you can have optimal productivity with just a green cover of the ground without anything growing high. The reason that trees are growing is because of competition and not necessarily to be more productive. Okay, so now we're talking about urban trees and urban environments. And we're all planting trees because it's getting hot and we want to be in the shade and we want that cool shade of the trees. And we're wondering about the importance of latent energy dissipation, right, in the energy balance. Where's the
Red thing.
red thing? Okay. Okay. So we're talking about latent energy dissipation in urban environments and whether the trees are more useful than just a passive shade structure. So this is part of a project with a group from Germany and Israel, and actually we have other people in it. And Wolfgang's name isn't here, and I'm sorry, because he gave us a lot of technical support. Now he's photographing us, which is also good for us. And the main people in this that worked with me on this, and the stuff that I'm going to present is Limor Shashu Abar. Background is from urban climate and stuff like that. I'm more of an environmental physiology, and Mohammed Rahman from the German group, who is actually a Pakistani. He wanted to get here, but didn't get a visa. Okay, so these are the institutions involved, and you can see there's a number of groups, and the funding was from a big DFG grant from Germany. We had Palestinian and Jordanian groups involved also, but not in what I'm going to be presenting. So there's a group from Munich and a group from Israel, and the name of the project is to get an idea of the impact of trees on urban microclimate in temperate Mediterranean major cities, from measurements to mechanisms and ecosystem services. And I won't read the rest. If anyone's fast, they can read it, but lunch is waiting. Okay. So let's take a look at the study sites. We were comparing two climates, and these are trees in Munich, and we called them by this name of tree labs, or basically a few sites in the city where we're setting up a lot of equipment to measure tree performance and how the trees are feeling. And here we have 3 sites in Munich, one with completely sealed surroundings. We have these trees growing and the ground is covered with bricks. And here's let's say more park setting. And then we have trees growing along the street, okay? And our counterpart in Beersheba, okay, which is, if anyone's not familiar with Beersheba, it's about 200 millimeters rainfall in a very, I'd say, the edge of the arid part of Israel, a little north of here. And this is, these are Prosopis alba trees, which is Yanbut in Hebrew, at the Sami Shamoun College. This college has about half a hectare of park area where the students like to sit around. It's very comfortable, even the very hot and arid summer, very pleasant under these Prosopis trees. And we have some near the street and we have some in this park area. Okay, so here's an aerial photograph of that site. It's a very interesting building built by a very innovative architect. This is the Sami Shamoun College. And here's the park site that we were working at. Here's the Prosopis trees. And here's some other trees, Tipuano Tipu trees along the street and some of the Prosopis trees along the street. Okay, and there's Bialik Street for any of the Israelis from Beersheba. And here's the satellite photo of Beersheba. You can see that it's a very arid region, and this is where the college is in Beersheba. Okay, so now, of course, here we have Wolfgang, who's on the ladder here, setting up the meteorological equipment. And of course, because I'm in the project, I'm putting some sap flow sensors, and we didn't have a lot of trees, so we stuck 2 sensors in each tree. And, okay, and we did about 2 years of measurements. Here's one of the trees, a big tree that's along the street. And let's take a look at the climate for a minute. So let's see the vapor pressure deficit. We have 2 sites in Munich, and SCE is the Beersheba site. Okay, so you see this is the summer in the beginning of August, and we have the VPD in Beersheba, which is getting up to about 4 kilopascal, and in Germany, in Munich, it's about less than 3. So we have a clear difference in the evaporative potential, and here's the solar radiation, of course, much higher in Israel than in Munich. Okay. So this is a contrast of a very arid area with a kind of temperate area in Germany. And here's the sap flux density. For anyone who is not familiar, so sap flux density is the flow rate in the stem relative to the stem area that's transporting it, right? The hydraulic area of the xylem, okay? Sap flux density in millimeters per centimeter square per hour. And here's in Munich, so there's Norway maple, I think, and Tilia cordata is a plane tree, and this prosopis trees, and you can see the daily curves. And this is end of July, beginning of August, when we have some nice data from both sites. So you can see there was a rainy period here in Germany. Of course, in Israel, the summer is completely rainless. And here we have the averages of let's say, of this period. For this, it's the rainless days. We didn't include the rainy days. And we see these typical daily curves. Okay, and here's the street area. In the park area, we suspect that the soil in the park area was very poor, and maybe there was some concrete underneath the trees. There's a problem in the urban areas. Anyway, so the trees in the park area were doing less than there. And let's take a look at these again. Let's take a closer look at these daily curves, and we see, first of all, we have similar sap flux densities in both places. And we know that more or less the amount of stem that's active is about the same. So these trees in both areas, different species, are doing about the same sap flux. And we have next to the street, we also have this kind of midday plateau for the highest flow rates, which is something that we've seen very often in our sap flow measurements. And here's from other projects. Here's from 4 agricultural species: apples, grapefruits, persimmon, and nectarine. We have this kind of summer data, the daily course of sap flow, which gives us very often this plateau, where we have for many hours, we have about the same at maximum sap flux densities. And the height of this, let's say the amplitude of these lines depends on the water potential. This is the midday and water potential. Okay, so this is a feature which I call isohydric. It means that the tree, when it gets to a certain evaporative demand, it reaches a maximum. It reaches a hydraulic limit. It reaches a water potential that it wants to stay at. And this is described, this is a nice review paper from Barbara Bond, Meinzer, and Brooks that describe this. Okay, we have the stomatal conductance as a function of increasing vapor pressure deficit or decreasing humidity, and the stomata close and transpiration kind of levels off according to the soil moisture, which in my case was the midday stem water potential. So we have this kind of leveling off, which is for me represents the hydraulic limit, and it's in the discussion of water deficits for isohydric plants. Okay, so this is that figure again, and we have this nice isohydric behavior in both climates for these trees. How much time do I have? Okay, I have another.
Oh,
Okay, good.
sorry about that.
Okay. Well, we've worked on this before. This is also from that paper. What comes out of that response is that constant sap flux density at midday comes out as this stomatal conductance curve that we saw before, and the stomatal resistance or stomatal canopy resistance will have a linear relationship to VPD. That comes out of having constant sap flux density at midday. And this is where we did it for, let's say, in apple trees on 3 rootstocks, we got the same kind of relationships. In 2002, we published that. Okay, so summary so far. Climate in Beersheba is hotter and drier than Munich, sorry. And sap flux density is similar. That means that the relative contribution of the trees in Beersheba is going to be less than in Munich, right? The cooling relative to the energy budget or to the evaporative demand is going to be less. Now we continue to convert this to a canopy projected area. We want to get a one-dimensional resistance for canopy conductance. Okay, and here we go. Let's take a look at the table of tree morphology. And let me just summarize this. We have here ground area, and the trees in the north, if we think about it, the trees in the north are growing really high. They're relatively narrow. That's what we're used to. And the trees in the south, in the arid area, are really low and wide. Okay. They're really shading out. And this is a kind of, let's say, a trade-off between, let's say, hydraulic efficiency for these tall trees and competitive efficiency of the wide canopy that's shading out everything else. Here's an example of one of the prosopis trees, untouched. Okay, we have this small stem supporting a very large canopy, and it's very wide and not so tall. And this has actually been published in at least I found at least 2 allometric papers that pointed out these differences between trees in temperate versus arid areas. And we also saw it in in the previous 2 presentations, these wide trees in these arid areas. Okay, so as I noted, the canopy resistance to VPD relationship comes out as a linear curve. And now, because we've taken these very wide trees, okay, with the large canopy projected area compared to the relatively narrow trees in Munich, and now we have large differences in the canopy resistance. And here we see the high canopy resistance, and let's say I'd call it the hydraulic resistance. We can show that this slope is related to the hydraulic resistance of the trees in Beersheba versus the low hydraulic resistance of the trees in Munich. Here is to summarize this. This is the sap flow, the average midday sap flow in watts per meter squared. Okay, and we can see the 2 points for Beersheba for the arid area, and the higher sap flow per unit ground area of the trees in Germany, in Munich. Okay, and this we have 2 other ways of presenting this relationship: the canopy resistance and the slope of this canopy resistance to VPD curve. Okay. Okay, I have, let's say, 2 minutes or so. Or I'm out of time.
2 minutes was fast.
Oh, that was 2 minutes?
Yeah.
So we see the difference here in the energy budget, and we see that the large difference of the low latent energy relatively in Beersheba to the higher latent energy of the trees in Munich, and compared to the differences in the net radiation that we can compute for these forest areas. Okay, and this is just the same. So my summary and conclusions. So we measured the canopy sap flow, and we had similar sap flux densities. The trees in Beersheba, the morphology was lower and wider, and in the bottom line, takeaway is latent heat flux per canopy area was lower in Beersheba, okay? Because of the net radiation is higher, and the relative contribution was less in Beersheba than in Munich. So this has implications for urban climate and for the trees that we're planting in the urban climate. Okay, and just some pictures of the participants. Thank you.
Yeah.
Yes, I have a question. Thank you. It was very interesting. I was wondering, so the trees that are wider and lower versus the trees that are taller, do you think they were actively managed or chosen because of that? Because you like to have shade in the desert, or do you think it has to do something with the environmental conditions?
Yeah, well, there's the difference in morphology of these species. As I noted, people that have studied the allometry of arid area, semi-arid area trees versus or semi-arid species versus more temperate species have found this difference. So, and this is, in my opinion, it's a trade-off. To grow tall means you're going to lose energy because you have to have a low the water potential gradient. And we know that that's what limits the tree size, is the water potential of these tall trees at the tips, okay? And where if you only need to shade out your neighbors, right? You want to grow a very wide canopy, and they really don't care about the efficiency for photosynthesis. You want to just maintain it green and just shade out everything. And that's the strategy in these arid areas.
If I'm an urban planner, I would also want to max getting cooling.
Isn't that true? Yes, definitely. So what trees are we planting? Okay, and are the planners familiar with this? So they're planting prosopis because that's what we grow, what grows in this area. But now we're starting to bring in the taller oak trees.
A lot of allergy.
What's that?
Allergy that is coming from the flowers.
Okay.
But it's
So
really.
now, so that's other ecosystem services and things like that.
With the plants in Beersheba, you had the subflow. Can you show it again, please?
Yeah.
Yeah. One of them was showing a uptake after a sunset. Pretty much quite a lot, and in the park was not. So can you show why is there difference? Is it what soil water content or?
And I'm not sure. I'm not really ready to answer that question. I'm not prepared, but what I can say is this is typical of what I've seen. As I showed for a number of other species that we've studied and more intensively. Each one of these curves is probably an average of 10 trees over a period.
Most of them you see after 8 o'clock there is almost zero subflow, and in Beersheba you still see after 8 o'clock water uptake.
Sometimes we've seen that, refilling.
We see it in day palms after until almost midnight. Yes.
We saw it in an irrigation experiment with corn once. We had a lot of recharge. Depends on the capacitance of the tree. But I don't have data for this.
Yes. Keeps you away from your lunch.
No, just a quick one. What was the delta temperature in the two sides below the canopy?
Below the canopy?
Yeah.
I'm sure it's about 10 degrees difference.
Strong advection.
Between, in the same, the two sites in the same
What was the
city?
delta in Munich and in Beersheba? For the same?
Between Munich and Beersheba?
What was the delta in Beersheba between shade and outside? And in Munich, it was the same?
Air temperatures are very similar. There's outdoors, we have a lot of mixing of the air. Air temperature differences should be more than 1 or 2 degrees.
Because there's some claim that capture the less turbulence when you have a white canopy, so less mixtures and the temperature is
But it's all turbulent anyway. As long as you're outside, it's all turbulent.